Assalamualaikum warahmatullahi wabarakatuh. Let me introduce myself. My name is Umi Ba'dingatul Latifah, and today I will be presenting a topic for the course Clinical and Forensic Analysis under the guidance of Professor Dwi Siswanta M.Eng, PhD. Okay, to continue my presentation, let me share my screen and um, explain about my PowerPoint. Okay, this is my PowerPoint, and the title of my presentation is Electrochemical Paper-Based Analytical Device or EPAD using Immobilized Prussian blue and antibodies for the diagnosis of Leishmania. Okay, the main reference for my presentation is research article published in ACS Omega in 2023, entitled Electrochemical Paper-Based Analytical Device or EPAD using Immobilized Prussian blue and antibodies for the diagnosis of Leishmania. This paper was written by Mauricio Alberto Poletti Papi and colleagues. Okay, and this study focus on the development of a qualitative electrochemical paper-based device that utilizes Prussian blue and specific antibodies to detect Leishmania antigens. Okay. Now that we know the main reference and focus of the study, now let's move on the clinical problem that motivate the development of this device, which is the challenge of diagnosing leishmaniasis effectively, especially in low resource setting. Leishmania is a zoonotic disease caused by Leishmania parasite and transmitted by female sandfly. It mainly affect a human and domestic animals such as a dog, and its epidemiology is influenced by environmental and migratory factors. The second point is transmission of leishmaniasis. Parasite transmission to human occur by the bite of an infected sandfly. The sandfly becomes infected after biting and parasites host. Person-to-person transmission by sexual contact, blood transfusion, and the use of contaminated syringes has also been described, but they are exceptionally rare. Okay, the Leishmania protozoa, a single-cell infectious organism, is usually spread through the bite of an infected sandfly. A person may experience mild or no symptoms or develop skin ulcers or cutaneous leishmaniasis, and then or ulcers inside the nose, mouth, or throat that can cause severe visual impairment, or mucosal leishmaniasis, or experience fever, weight loss, fatigue, and an enlarged spleen and liver. It's called visceral leishmaniasis. Doctor diagnosis infection by analyzing a sample of infected tissue or performing a blood test. And the third point is clinical form of leishmaniasis. There are three. The first is cutaneous leishmaniasis, or CL, is the most common form and causes skin lesion, mainly ulcers on exposed part of the body. This can leave lifelong scars and cause serious disability or stigma. And number two is mucocutaneous leishmaniasis, or MCL, leads to partial or total destruction of mucous membrane of the nose, mouth, and throat. And the third is visceral leishmaniasis, or VL, also known as Kala-Azar, is fatal if left untreated in over 95% of cases. It is characterized by irregular bouts of fever, weight loss, enlargement of the spleen and liver, and anemia. Okay, leishmaniasis is a neglected tropical disease prevalent in developing and underdeveloped countries, with up to 1 million new cases each year. Leishmaniasis is a well-known disease, appearing mainly in the countries of Southern Europe. It is an endemic zoonosis in 88 countries around the world. Although 90% of affected individuals are located in 5 countries. There are India, Bangladesh, Nepal, Brazil, and Sudan. According to the World Health Organization, the annual incidence is estimated between 1.5 and 2 million cases. The prevalence is 12 million, and the population at risk of being infected is estimated at 350 million people. World Health Organization Technical Report Series 949 in 2010. Okay, now is the clinical problem, conventional diagnosis. Conventional diagnosis like parasitology, serological tests like ELISA, and Western blot has drawbacks. There are long-lasting times, high costs, and limited availability in specialized laboratories. So, the urgency of this research is limitation of current diagnostic method, restrict healthcare access in remote and economically disadvantaged region. Number two is there is a need for rapid, low-cost, sensitive, and user-friendly diagnostic tools to improve early detection and management of leishmaniasis. And the last is biosensor, especially paper-based electrochemical immunosensor, or EPET, offer portable, affordable, and field deployable solution. So the research objective, the first is to develop and evaluate an electrochemical paper-based analytical device EPET for the qualitative diagnosis of leishmaniasis. The second is to provide a rapid, low-cost, and accessible method for detecting Leishmania amazonensis antigen with good performance and high selectivity. And the last is to demonstrate the proof of concept of EPAT as a relevant diagnostic tool for neglected disease in underserved region. Okay, go to next slide. This is about principle and method. So the first is biosensor or device working principle. The electrochemical paper-based analytical device or EPAT detect Leishmania antigen using a sandwich-type immunoassay. And then specific antibodies and HRP enzyme are immobilized on paper, capturing the target antigen and generating an electrical signal through Prussian blue redox reaction. And the material or technology used in this research is the first substrate filter paper disc and Prussian blue being mediator, and anti-Leishmania antibody is bioreceptor. Detection label is HRP or horseradish peroxidase, and screen printed carbon electrode and portable potentiotat system. Okay, this image shows the preparation step for a filter paper disc used in the EPAT biosensor. The first filter paper is cut into a small disc and then clean with a chemical solution using ultrasound and rinse again until the surface is clean. Next, mediators such as Prussian blue are applied to the surface, and the paper's surface is then chemically activated so that antibodies can be effectively immobilized. And then this process ensures that the paper is ready to be used as a sensor platform for clinical antigen detection. Okay. This image illustrates the workflow for detecting Leishmania antigen using the paper-based EPAT. First, a captured antibody is immobilized on the paper surface. If the patient sample does not contain the antigen or negative section, the detection antibody does not form a sandwich complex. So there is minimal enzymatic reaction and the current remains high. In contrast, if the sample contains Leishmania antigen or positive section, a sandwich complex form and the HRP enzyme catalyze a reaction with H2O2, causing a significant decrease in the measured current. The difference in current between positive and negative sample is the basis for diagnosis. So, the clinical target of this research is the first biomarker analyzed Leishmania amazonensis antigen or synthetic peptide proof of concept model. And the second is relevance of biomarker for clinical diagnosis. This antigen is specific for diagnosing Leishmania amazonensis infection, which causes leishmaniasis. Detecting the antigen signifies an active infection in patient. Okay. The next point is about normal versus abnormal concentration range. The EPET study for detecting Leishmania antigen did not establish a numerical range for normal or abnormal concentration because the sensor is designed as a qualitative rather than a quantitative detection tool. The standard for interpreting result is based on the presence or absence of the target antigen is the patient serum, as indicated by the amperometric response detected by the sensor, rather than a concentration value. Normal or negative sample that do not contain Leishmania antigen. No sandwich immune complex is formed, resulting in a high mixture electric current. The amperometric graph approach the baseline because the consumption of H2O2 by HRP is very low. Abnormal or positive sample where Leishmania antigen is present in the serum, a sandwich complex is formed, the HRP enzyme become activated and catalyzes the H2O2 reaction extensively, causing the measured electric current to drop sharply. The signal decreases compared to the control. Okay, now is about my main result. The research summary is the research developed an electrochemical paper-based analytical device or EPET for the qualitative detection of Leishmania amazonensis antigen. The device uses Prussian blue and specific antibodies immobilized on paper. Operating via a sandwich immunoassay with HRP label, antibodies, and hydrogen peroxide. The EPET showed good reproducibility and selectivity, providing a rapid, low-cost, and portable diagnostic solution for point of care use in underserved regions. The next is sensitivity, selectivity, limit of detection, and linear range. The device achieves a clear distinction between positive and negative sample with a 95% confidence interval. And then the limit of detection, or LOD, was not explicitly given a numerical form due to the qualitative nature of the sensor. It showed excellent selectivity with no interference from yellow fever virus in tests. And then the immunosensor optimization was done with capture antibody concentration as low as 100 microgram per milliliter and peptide concentration in the range of 100 microgram per milliliter. Optimized through factorial design. The relative current drop correlates to the presence of the Leishmania antigen and demonstrating effectively sensitivity. So this is the visualization of the data, and the first figure is shows scanning electron microscope or SEM images illustrating the step-by-step modification of the paper substrate used in the development of the EPAT biosensor. There are panels A, B, and C display the untreated paper fibers showing their natural, unmodified structure and porous network. There are panels D and E show the paper after modification with Prussian blue and subsequent immobilization of specific antibodies. You can observe changes to the surface, including coverage and new texture compared to the raw paper. And panels F and G show the final biosensor surface after completing all modification steps, including the attachment of biomolecule and possible blocking agent. In panel G, yellow arrow highlight the presence of immobilized nanoparticle or biological element, confirming successful functionalization of the paper. These same images provide visual confirmation that each modification step was successful, and that Prussian blue and antibodies were evenly distributed on the paper surface. This support the device ability to detect the target antigen through effectively signal transduction. So the second picture is both of these figures are visually confirmed that the EPAT can reliably distinguish between negative and positive sample through a clear and reproducible amperometric signal. Okay, and the third figure is demonstrates the selectivity and performance of EPAT biosensor for detecting Leishmania antigen in various sample types using amperometric signal percentage. Okay, so there is the table is comparison between biosensor applied for the detection of Leishmania. Okay, the table is present a comparison of various biosensor that have been applied for the detection of Leishmania. The table summarize information about each biosensor detection technique. The biological target is it recognized, such as antigen, antibody, or DNA. The type of sample it analyzes and how its selectivity has been validated. For example, the table shows biosensor that use technique like cyclic voltammetry or CV, and then differential pulse voltammetry or DPV, electrochemical impedance spectroscopy or AES, and multiple pulse amperometry or MPA. Each biosensor is tested with different types of sample, including human and animal blood, serum, or even human biopsy specimen, and the selectivity analysis involve testing against other pathogen or non-complementary sequence. The last row describes the IPET SPCE developed in this work, which uses MPA to detect Leishmania amazonensis peptide in human blood serum. With yellow fever virus used for selectivity testing, and this comparison highlights the strength and specific application for each biosensor in different diagnostic contexts. And then the method advantages in this research is low cost, simple material, and then suit for resource limited regions. The second is high portability, small, easy to use without specialist lab. And then reproducible and selective. Does not require a special electrode modification, making it cheaper and more versatile. So this iPad is suitable for point-of-care services. And the limitation of the method is provide only qualitative detection, no precise quantitative data, and then non-specific absorption and sample matrix variation may cause interference. And the third is sensor durability for a use remains suboptimal. And the last is long-term stability has not been tested in real-world environment. So, the clinical realism for this research is highly promising for rapid screening, but further confirmation is still required for definitive diagnosis by standard method like PCR or ELISA. And then the implementation in hospital or clinical lab is suitable for point-of-care use like health center, village clinic, and field screening. So this is support early detection, especially in underserved facilities. And the relevance to patient is faster and more affordable diagnosis, increasing healthcare access, and then reduce complication and delays in therapy initiation. So, future development direction. The first is development of multiplex biomarker detection. And number two is adaptation for other tropical disease or chronic infection. And the last is enhancement of quantitative future, sensor regeneration, and system automation. Okay, and the last is conclusion. IPET for Leishmania is proven to be sensitive, selective, and highly promising for screening in limited resource area. And simple application, low cost, no need for complicated equipment. And the last is initial evidence support field clinic, and remote area usage. So the final message is paper-based biosensor innovation opens opportunities for fairer, simpler, and faster detection of neglected tropical disease for high-risk communities. Okay, this is the bibliography that I use in this review article. And the last is, thank you. This is enough for me. Wassalamualaikum warahmatullahi wabarakatuh.